Great to be talking to you guys today. So I'm going to be presenting about half of my thesis research to you today, and that focuses on the noradrenergic modulation of experience-dependent plasticity in auditory cortex. So I study primary auditory cortex, and that is a subregion within the larger auditory cortex, and it's the first relay that an auditory signal hits when it reaches the cortex. So as such, it has some response properties in common with lower auditory areas. It responds to the frequency, duration, intensity of the sound. But it's also starting to do some higher order functions like pulling out the meaning of the sound. But the salient feature of primary auditory cortex that I want to focus on for the purposes of this talk is its tonotopic. So neurons in primary auditory cortex have a particular frequency that they respond most strongly to, and that frequency dictates their spatial organization in primary auditory cortex. And tonotopic is hardwired to some extent. So even if you have no sound experience, this property will arise. But it can be modified with experience during a period of development. And so what that would look like is if you, say, had a lot of experience with 4 kilohertz tones for some reason during development, that area of your brain, called primary auditory cortex, will become larger at the expense of neighboring areas that respond to frequencies less common in your environment. And this is a great example of a developmental sensitive period. So you've probably heard of this before. The best known examples are in the visual system and language system. And the salient things about sensitive periods for the purposes of this talk are during development, the changes that occur during the sensitive period will persist into adulthood. So they're long lasting. And during adulthood, simple exposure is not enough to drive this kind of frequency expansion, at least in primary auditory cortex. You need to pair that with some sort of salient event, a reward or a punishment or something like that. And so I became interested in this. I wondered why is it easier to reduce plasticity during development than in adulthood? And there's not a lot of research into this in auditory cortex, at least. So I had to kind of look into other fields to point me in the right direction. And there's more research in the mechanism behind sensitive period plasticity in the visual system. So the classic studies of Kugel and Wiesel in the 1960s kind of point to this. And since then, there's been a wealth of research into the mechanism of the sensitive period visual cortex. And what that research suggested to me is that norepinephrine might play a role. So norepinephrine, as a lot of you probably know, is produced in locus coeruleus in the brainstem. And that's the sole supply of norepinephrine to the cortex in the forebrain. So it's really kind of diffusely throughout the forebrain from locus coeruleus. And norepinephrine is a neuromodulator, so it's not necessarily going to cause neurons to fire, but it will increase or decrease the likelihood of neural firing, modulating responses to stimuli. So the research I mentioned in the visual system suggests that norepinephrine might be required. You have to have it on board in order to see sensitive period plasticity in visual cortex. However, this was disputed a decade after that initial finding by another lab which said you need either norepinephrine or acetylcholine, but you don't need one or the other definitively. And since then, this issue has still kind of been controversial. There's really, no one's landed on the answer of whether it's required or whether it just plays a role. And I think that's partly because the methods that were used to lesion the noradrenergic system in these studies were these neurotoxic lesions. So you can inject a toxin that will target and kill the neurons that produce norepinephrine. And these methods can be notoriously incomplete or nonspecific. They can affect other modulatory systems in addition to the noradrenergic system. So I knew that if I wanted to investigate this question, I would need to find an animal model that produces a better, more clean lesion of the noradrenergic system. Fortunately, that's available to me here at Emory. David Weinschenker's lab rears a colony of dopamine beta hydroxylase knockout mice. So these mice are missing this enzyme, dopamine beta hydroxylase. This is a norepinephrine synthetic pathway. So without this enzyme, they can't make norepinephrine. So this is the experimental group I decided to use. And for controls, I'll be using their littermate controls. They're heterozygotes. They have one allele of the gene for PBH, and they have normal levels of norepinephrine. So these are my 2 genotypes that I use, but then within that, to really test the idea of sensitive period plasticity and the role of early experience on frequency responses in primary auditory cortex, I needed to do sound manipulation. So within each of these genotypes, I expose animals to 2 different sound environments during development. One group is just a baseline group, so they're born and then they just live in a colony until adulthood. The other group is a sound exposed group, and this is a group that I exposed to repetitions of 8 kilohertz tones for a 2-week period during development. So to give you an idea of what that sounds like, I should be able to play this. So they're just hearing that for like 16 to 24 hours a day. That's how they spend their childhood. So it's not a subtle stimulus, and it's one that's been shown in the literature previously to drive the sensitive period plasticity during in adulthood. So you can see the effect of this in adulthood. And to test whether I was successful in driving sensitive period plasticity in these animals, I would do a mapping experiment when these animals hit adulthood. And so before I go on, I'm going to go into some detail now about how I do these mapping experiments. So when I do a mapping, I'm trying to construct a frequency response map of the auditory cortex. And the first step I have to take is to open up and access the primary auditory cortex. So I do that by opening up the craniotomy on the left auditory cortex in this mouse. And this is about a 3 millimeter square kind of hole in the skull. It exposes the primary auditory cortex, and then I'll drop an electrode in to record neural responses there. And this is what my electrode looks like. It's actually 3 of them bundled together. But for the sake of example, I'm just going to walk you through one sample recording site. So I put my electrode in the brain, and I'm recording local activity of a population of neurons around that electrode. So not just one neuron, but a whole bunch. And I'm recording a voltage signal that looks like this. So it's showing you voltage over time. And while I'm recording a signal, I'm presenting tones to the animals, and that's what this black bar represents. And you can see for this recording site, shortly after tone onset, the voltage starts to fluctuate, and it's showing the neurons firing. I do this at, I play tones of many different frequencies to the animal. And then, oh, I'm sorry, so I'm going to threshold, after I do this recording, I'll threshold the voltage signal. So every time the voltage crosses this threshold, and we count that as a spike. And then I'll do this after presenting a number of different frequencies to construct something called a tuning curve. So a tuning curve is showing you the firing rate in spikes per second over the different frequencies that I play back. So I'm playing back frequencies from like 2 to 30 kilohertz. And as I said earlier, neurons in primary auditory cortex have a favorite frequency that they respond most strongly to, and that's clearly here about 25 kilohertz. So we'll label that the best frequency. Once I know the best frequency, I can color code this site, and then I'll repeat this process over the entire spatial extent of primary auditory cortex and construct this. This is the map of the frequency responses. And so this is a representative map from an experiment I did last year. And this shows you primary auditory cortex, which is about from like the halfway point here, so caudally. And the mouse also has another primary-like area called the anterior auditory field, and that's here. So it's like a mirror image of primary auditory cortex. And these are both sites at which I might expect to see plasticity, so I've included them both in my analysis going forward. Okay, so after I constructed maps of the 4 different groups that I described earlier, and before we even get to the question of what sound exposure is doing in the presence or absence of norepinephrine, you could wonder what's happening at baseline. It's possible that norepinephrine is involved in so many different processes, you might see something different just at baseline, even in the absence of sound experience. So the first step I was interested in is there a difference just at the outset? And so these are representative maps from my knockouts here, and then my norepinephrine competent controls. And these maps look slightly different, but that's just a reflection of individual variation. I did this for about 9 animals per group. And then if you construct an average, and then I can kind of average these maps all together to create this figure. This is like the average map for each group. And then it becomes pretty clear that there's no obvious difference. So there's no overt disorganization in the knockout animals, no rotation in the cortex. And then we can quantify this by making a cumulative distribution. So this is showing you the number of units here on the y-axis that respond, have best frequencies at all of the frequencies I tested here. And the shape of these curves is not significantly different. So this made me pretty confident that at baseline, norepinephrine is not doing anything to predispose animals to have different sort of cortical organization. This provided me with a really nice baseline from which I can task my experience-dependent question. What is going to happen when these animals are sound exposed to 80 kilohertz during development? So before I go forward, okay. So this is, to give you an idea of what I expected to see, we know here that these are starting out the same. So this is kind of a dummy map showing you that these animals are the same at baseline. I knew what I would expect to see in the heterozygotes in the sound-exposed animals. These are just wild-type animals. Other work has shown that you'll see more responses to the exposed frequencies. So that would be this greenish color that corresponds to 8 kilohertz. The real question was, what would I see in the knockout animals? If norepinephrine is not required for sensitive period plasticity, then their map would look pretty similar to the heterozygotes. I'd be able to drive an increase in the expression of the 8 kilohertz frequency. If it is required, though, I would not be able to drive an expansion in that frequency, and this map would look more like the baseline map from that group. So that's the big question. And I'm going to show you what I've seen so far, but noting that this is still underway, the study's still underway. I have about 3 animals in this group and then 8 or 9 in the rest, so I still need to add animals here. But just preliminarily, what I've seen so far, in the heterozygotes, so this is just showing you just the percent of cortex tuned to 8 kilohertz. And these are the heterozygotes who have norepinephrine, normal levels of norepinephrine. And in them, I saw exactly what I expected to see, about a doubling in the representation of 8 kilohertz following sound exposure during development. And this effect size is totally consistent with what others have observed in the literature. And this is great. This shows me that my paradigm is working the way I want it to work. In the knockout animals, though, so far I haven't seen a similar increase over baseline in the sound exposed animals. So you might notice here that the baseline levels look somewhat different, and although that's non-significant, what's important is that you're not seeing an increase over the baseline in the sound exposed group. So far I have not been able to drive sensitive period plasticity in norepinephrine deficient animals. So to give you an idea of what that means, if this is borne out over the entire population of animals that I plan to map, this would suggest that norepinephrine is necessary for sensitive period plasticity, at least in auditory cortical development. And my hope is that it would go on to motivate studies of visual cortex to help kind of settle the issue there. Now that we can take advantage of these better genetic techniques that allow a cleaner region of the noradrenergic system. What does this mean for development? Even if norepinephrine does turn out to be necessary for sensitive period plasticity, it's likely working in concert with other neuromodulators. So more studies would need to be undertaken in serotonergic systems, acetylcholine, which is has also shown to have a role in visual cortical development to kind of illustrate the full complement of neuromodulators that contribute to this effect. And then this is a pretty basic level study. This is sort of reductionistic, but to give you an idea of the translational relevance of this work, I wanted to end on an example of work that a colleague in our field is doing that sort of takes advantage of this idea of neuromodulation to reopen a period of plasticity in the mature cortex after development has concluded. So I'm going to start by talking about vagus nerve stimulation. So vagus nerve stimulation is a treatment for intractable epilepsy, and it works by they just insert vagus nerve is a cranial nerve, so you can insert an electrode just under the skin, kind of over the sternum, and stimulate the vagus nerve, and that alleviates epilepsy. And it's believed to work through neuromodulatory systems. So this is a schematic of how vagus nerve stimulation acts. So the vagus nerve is down here. It synapses mainly on the nucleus of the solitary tract. And then that works through the locus coeruleus, releasing norepinephrine into the cortex and forebrain areas, and the nucleus basalis, releasing acetylcholine into forebrain areas. And those are believed to kind of dampen the rampant excitatory activity that leads to seizure. So if this is kind of a relatively more naturalistic way of increasing neuromodulatory tone in the cortex instead of just dumping a lot of extra on. This kind of harnesses the body's endogenous mechanisms for releasing. So as I said earlier, a colleague in our field has kind of seized on this and recognized that this might be able to promote plasticity in the cortex in a therapeutic way to alleviate an auditory cortical disorder called tinnitus. So you've probably heard of tinnitus, even if you're not sure exactly what it means. It's ringing in the ears. And usually it occurs at one particular frequency. So for the sake of consistency, let's say 8 kilohertz. You have tinnitus at 8 kilohertz. Well, they've created a rat model that these rats will have tinnitus. And they've stimulated their vagus nerve and paired that with tone presentation in all of the frequencies surrounding the tinnitus percept. So if it's at 8 kilohertz, they'll pair vagus nerve stimulation with other frequencies outside the 8 kilohertz range. And by doing this, they're able to kind of renormalize the representation of 8 kilohertz in the tinnitus auditory cortex and eliminate the tinnitus percept. And this is currently in clinical trials now in Europe, actually. So it remains to be seen whether it will work in humans. But if it does, this is the type of sort of overall relevance that this neuromodulation plasticity, I hope, will have. down the line, but we're still a ways from that yet. So that ends the science portion. I just wanted to take a minute to thank everyone who's helped me complete this work. So first off, my advisor, Robert, who has been super supportive and enthusiastic and has just set a great example for me. My whole lab, and this is us, kind of Bontrap family style. And they've been great in helping me kind of talk out my ideas. And then the wine checker lab, which has supplied me with mics and helped me kind of hammer out some of the neuromodulatory aspects my lab is less familiar with. And then this has been supported by NRSA, and then I was also supported by Emory's Prism Fellowship, which is a teaching fellowship offered here at Emory, and with some help from Robert also. And finally, the Emory Neuroscience community, which is, as people, I'm sure people have been telling you all morning how great a place it is to study neuroscience, and that's certainly true, but I'd like to point out it's also a great place to live your life outside the lab, and I know I couldn't have done it without all the support of our community, so thank you guys for coming out. I'm glad to be talking today.